How does SCNT molecular biology make reproductive cloning possible? Imagine being able to create a living copy of a person or animal using only a cell from their body. That's what somatic cell nuclear transfer, or SCNT, makes possible. It's a process that involves moving genetic material from one cell into another. This technique is the key to reproductive cloning, which aims to produce a new organism that is genetically identical to the donor. The process starts with removing the nucleus, which contains most of the genetic information, from an egg cell. This step is called enucleation. Then, a somatic cell, like a skin cell, is taken from the organism you want to clone. The nucleus from this cell is carefully inserted into the enucleated egg. This is the nuclear transfer part. Once the donor nucleus is inside, the egg is stimulated to start dividing as if it had been fertilized naturally. Inside the egg, factors in the cytoplasm reprogram the somatic nucleus. They erase many of its specialized marks, called epigenetic marks, which are responsible for cell identity. This reprogramming restores the nucleus's ability to develop into an embryo. It involves changes in DNA methylation and chromin structure, which are ways cells control gene activity. Sometimes, these changes aren't perfect, which can make cloning less efficient and cause abnormalities. Once the egg begins dividing and reaches the blastocyst stage, it can be implanted into a surrogate mother's uterus. If successful, the embryo develops into a full organism that is a genetic twin of the original donor. This is how Dolly the sheep was created in 1996, the first mammal cloned using SCNT. From an ethical point of view, SCNT raises questions about respect for human dignity and individuality. Cloning challenges ideas about what makes each person unique. It also brings up concerns about safety, as cloned animals often face health issues. The high failure rate and potential for abnormalities make human cloning ethically controversial. Justice issues also come into play, like who should have access to cloning technology and how it might be misused. In addition, the process involves manipulating embryos and genetic material, which raises questions about consent and the rights of potential offspring. While SCNT offers exciting possibilities for regenerative medicine and personalized therapies, especially through therapeutic cloning, its use for reproductive purposes remains a topic of debate. In essence, the molecular biology behind SCNT makes reproductive cloning possible by reprogramming a somatic cell's nucleus to an embryonic state. This ability to reset the cell's developmental potential is what allows scientists to create a living, genetically identical organism. But it's essential to carefully consider the ethical principles involved as we explore these advanced medical technologies.